In many cells, there's a discrete body which nucleates microtubules, and it is the centrosome. In other cells, we don't have the centrosome. Centrosome plays a very important role. As we have seen, it hosts a specific type of tubulin called gamma tubulin, which results in formation of new microtubules. So let's look at the structure of centrosome. Microtubules that are nucleated at the centrosome, they have their plus end pointing outwards towards the cell periphery. Centrosome is composed of fibrous centrosomal matrix, which is sort of an amorphous material that contains more than 50 copies of a gamma Turk, which is gamma tubulin ring complex. Inside this amorphous material, there are centrioles. They're cylindrical structures which are arranged at right angle to each other, forming roughly an L shape. And this structure is embedded in the centrosome. In motile cells, the centrioles, they become the basal bodies for cilia and flagella, which we talked about briefly in one of the previous modules. So here's the structure of centrosome. Here you can see there are a pair of centrioles. These centrioles are embedded in this amorphous material, which is surrounding these pair of centrioles. In this amorphous material, you have the gamma Turks or gamma tubulin ring complexes, which are the nucleating sites for microtubules. As I drew earlier, the minus ends of microtubules are attached to the ring complexes, and the plus ends are away in the opposite direction of the minus ends. So this centrosome basically ensures that microtubule array is organized. Not only it nucleates microtubules, it also organizes the microtubule array in many cells, and relative to this microtubule array, different organelles can be placed in the cell at specific location. Here you have a centriole. You can see the centriole. It is made up of microtubules actually, and the centriole is surrounded with this material. This is the amorphous material surrounding the centrioles, forming the centrosome. The centrioles organize centrosomal matrix, and which is also called, by the way, pericentriolar material, and also it ensures its duplication during cell cycle as centrioles themselves duplicate. The centrosome duplicates and splits into two equal parts during interphase, each half containing a duplicated pair of centriole pair. Microtubule nucleating sites are again distributed throughout this amorphous material. In plants, we don't have a centrosome, or they don't have the centrioles either, but they have microtubule nucleating sites distributed all around the nuclear envelope. So there's a difference as far as microtubule organizing center is concerned between plants and animals. In animals, the astral configuration of microtubule is very robust and dynamic. We just watched a movie which proves this point. Microtubules radiating out from the centrosome also survey the outer regions of the cell, and they can position the centrosome in the center of the cell. So you always see centrosome in the center of the cell. I will show you an experiment in the next slide, how centrosome is placed in the center of the cell. And relative to this position of the centrosome or its microtubule array, other organelles can be placed in different locations. It's organized. The way it is done, how cells place different organelles in different locations, it is done via help of motor proteins and also interaction of microtubules with membranes of the cell. The cytoskeleton, the centrosome, always finds the center. Let me show you this experiment. Here in a square well, a single centrosome was placed. It was placed on one side of this well, and microtubule subunits, which are basically alpha and beta dimer, they were added to these wells, and they provide appropriate conditions for microtubule polymerization. As you can see later on, as the microtubules started coming out of the centrosome, these microtubules started pushing the centrosome towards the middle. So it is an star-shaped array of microtubules, and these microtubules are polymerizing and depolymerizing, and ultimately they will, since all forces have to be balanced, ultimately this centrosome will move and will position itself in basically dead center of the square well. So organize the centrosome, and then you can organize rest of the cytoplasm relative to the centrosome. relative to, of course, the centrosome or the microtubule array. I'll show you another experiment. Here we have specific type of cell called melanophore cell. This cell, if we cut a part of the cell, here the blue arrows indicate the site where the cut was made with a needle. So you have from one cell, now you have two cells or a celllet. One is a complete cell, other it has certain components of the cell, but not the entire, or not all the organelles. Of course, it's lacking the nucleus. After a certain amount of time, what we see is microtubules organize themselves. These microtubules have a same organization as they are organized inside the cell, which is plus end towards the membrane, the cellular membrane, the plasma membrane, and minus ends towards the center. So how do microtubules organize themselves without the centrosome? They can use motor proteins and they can organize themselves in the same position if the centrosome was there. Although, of course, if there is no centrosome, you cannot nucleate microtubules, but you can still organize microtubules even if the centrosome is not present. Cells have other redundant mechanisms to make sure the microtubule array is organized properly, because everything else depends, all the rest of the cellular organization, everything else depends upon it.